Okay, so we are still talking about ways to control microbes in the environment. And we're going to talk first about the physical methods of microbial control. And there's a handful of these. They tend to center around concepts of heat, cold, filtration, radiation, things along this line. So things that are not chemicals. So physical methods are things that are not chemicals. And then the other part of the lecture will be chemical methods of microbial control. So heat-related methods, just a review. You remember that high temperatures denature proteins, disrupt membranes and cell walls, and disrupt nucleic acids. So really heat hits all of the main targets that allows us to control microbial growth. So it's a great method when it can be used. Fundamentally, there are two types of heat. We call them moist heat and dry heat. As you might guess, moist heat involves water or liquids, and dry heat doesn't involve any water. And we'll go over those in just a sec. What we're trying to accomplish basically is to have a method that can achieve the shortest, what's called decimal reduction time. Decimal reduction time is defined as the amount of time needed to kill 90% of cells at a certain temperature. So when you're figuring out how to do a heat-related method, you try to determine what's the shortest amount of time that and at what temperature can you get 90% of cells to be killed. So decimal reduction time is a measure of the speed of microbial death. This chart is the one this graph is the one I showed you earlier. It was used in that graph I was looking at a chemical version of microbial control, but in this case we're looking at heat. If you take a specimen, and the specimen has, let's say you start with 10 to the 4, it's almost 10 to the 4, 10 to the 4 microbes per milliliter, let's say, and you have, so 10 to the 4 would be 10,000 microbes per milliliter. Then what you would do is, at a certain temperature, you would heat it, and you would take a sample out every 2 minutes, maybe, and count how many microbes are in those samples, and then you would plot it. And what you would be plotting is the death rate. And what we look at, since each of these points on our scale, on our number of living microbes scale, is exponential, then the difference between any step to the next step is a tenfold difference. So the difference from 1 to 10 is a times 10. The difference from 10 to 100 is a times 10, and so on. So if you have a drop in the slope of your death rate that is equivalent to one of these divisions, then what we're saying is that if you have 1,000 organisms at this point, and a little bit later you have 100, then that represents one division drop in slope represents a 90% killing. So we call that the decimal reduction. And the time of the decimal reduction is equivalent to the x-axis, the x component of that slope. So in this case, I'm just going to randomly, I think it's more or less here. So let's say this hits around 11 minutes. This line hits at 6 minutes. So the amount of time at this particular temperature that it takes to reduce the number of living microbes exponentially, or by 90%, is 11 minutes minus 6, so 5 minutes. That would be the decimal reduction time. And since the slope is consistent all the way down, you could also measure this drop here and then measure the decimal reduction time. The easiest thing to do, though, is pick a point that wherever it hits there, and then look for the X component of that, basically the triangle that you're making. And that would help you to determine the decimal reduction time. And let's say that the decimal reduction time for this graph is 5 minutes, and let's say this is 80 degrees Celsius. If you increase the temperature, typically what's going to happen is you'll get a steeper slope, which will have a shorter decimal reduction time. So at higher temperatures, you'll get a shorter decimal reduction time. So moist heat is also used, moist heat is used. It's the most effective kind of heat. Moist heat is more effective than dry heat, which may be kind of surprising, but it's actually more effective. You get a quicker decimal reduction. You get a shorter decimal reduction time when you use moist heat compared to when you use dry heat. The things that are considered moist heat, you're probably familiar with the term boiling. You may be familiar with the term autoclaving. Autoclave is the machine. We have one in the prep area of our lab where you put different, that's how we sterilize media. It's how we sterilize test tubes, how we sterilize anything that needs to be sterilized, we typically put in the autoclave. And that involves not only heating, moist heat, but also high pressure. So boiling is good, autoclaving is better, but to autoclave something, you need an expensive piece of equipment. Boiling, for most purposes, boiling is enough. If you need to, if you have water that you're not sure is drinkable, you can boil it. And when the city puts out what's called a boil notice, when the city water supply puts out a boil notice, they'll tell you how long you need to boil the water before you can drink it. Of course, you let it cool and then you can drink it. And that sometimes happens if the city water supply gets accidentally contaminated somehow. So boiling is all about killing everything. The only problem with boiling is endospores. Some cysts and viruses can survive, so that's when you definitely need to go to autoclaving for those kinds of concerns. Pasteurization is a type of moist heat that's used for food products like milk and apple juice. I think they use pasteurization for that. That's kind of like a flash heating, but that is not meant to completely eliminate all microbes. It just reduces the total number of microbes. So what that means is the food product can still go bad, but it will last longer before the microbes are able to grow to a significant number. And then you have something called ultra high temperature sterilization, which is sometimes used for food products. There's actually some milk that you can buy that sits on the shelf at room temperature because it has been completely sterilized. But that's kind of a step beyond typical pasteurization. This picture just shows an autoclave. Some of you may have one of these if you work in maybe a dentist's office or some doctor's offices, or maybe even like a nail salon. They may autoclave the tools, and this is kind of a tabletop version of an autoclave. In a laboratory situation, you would have an autoclave that goes like, it's more the size of a large refrigerator. But they work the same. There's a very tightly closing door on the front. And this picture down here, you're looking at the side view so you can see inside. Once you close that door, and it has to have a lot of attachments so it stays closed, then there's steam and there's pressure. So boiling water and high pressure for about 15 minutes will basically sterilize almost anything. We do sometimes, if let's say we're trying to sterilize tools, surgical tools, and for some reason we think that we might have like some C. diff, for example, in the hospital, then, and remember, Clostridium makes spores. So we would want to, sometimes what they will do is they will put this little kind of gadget into the autoclave with the surgical instruments. And what this does is, if the autoclave is able to sterilize the surgical tools, it will be able to sterilize the spores that are in this container. And the container stays closed, but the steam is able to get in. And what happens is there's a color indicator, and if they pull out the everything out of the autoclave and this little gadget is red, it means the spores were killed. If the indicator turns yellow, it means that the autoclave did not reach enough heat or pressure to kill the spores that were in this little gadget. And so it just gives you an indicator, kind of a, hopefully you get this result here, and it gives you kind of an assurance that the autoclave is doing what you hope it's doing, which is sterilizing and killing endospores. So pasteurization is used for like milk and fruit juice and some milk products. Like I mentioned before, it's not sterilization. It reduces the amount of microbes, but eventually the food will spoil. It just takes longer. Typical pasteurization is 63 degrees for 30 minutes. You don't need to memorize that, but I just want you to see it doesn't boil. But if you do the ultra high temperature ones, then it does go above boiling, but only for a very brief period. It's like a flash pasteurization or a flash sterilization. And these kinds of things here will give you basically sterile milk. And you don't have to put it in the refrigerator. And they do sell that at grocery stores here, but I guess it just depends on what you're used to. But the stuff that you get out of the cold, the refrigerated compartment at the grocery store is just pasteurized at a much lower temperature. All right, dry heat. Dry heat is basically when you put something in your oven, that's dry heat. Okay, so let's say that you wanted to sterilize something, you could put it in your oven and, put the heat all the way up. And if you do it for a long enough period of time, that will very likely sterilize it. In lab, we sterilize our loops with dry heat, right? Because we put it right in the flame. If you burn something completely, incineration is kind of the extreme endpoint of dry heat. That's extreme. Incineration does sterilize. And dry heat does sterilize if you use it for a long enough period of time. But typically you need a higher temperature and a longer period of time than moist heat. So if you incinerate like a dead body, then the ash that is obtained from that is sterile because nothing can survive incineration. Okay, so it depends on what purpose you're trying to achieve, what kind of dry heat you might use. Now, in this case, we have refrigeration and freezing. This is just for microbial control. It doesn't kill stuff, but it stops the growth. But when you when you warm it back up, it'll start growing again. So metabolic reactions move slowly at low temperatures. When water turns to ice, it cannot act as a solvent for chemical reactions inside the cell. And so refrigeration stops growth, but you guys remember there are things called psychrophiles that can grow at cold temperatures. But anything else will typically stop growing or slow down growing at cold temperatures. And then desiccation means drying. Lyophilization means freeze drying. And both of those, by removing water, you cause the metabolism of cells to stop, but it doesn't necessarily kill. It just stops the growth.